Cellular respiration is a process performed by most cellular organisms, including animals, plants, fungi, protists, and the majority of bacteria, in order to convert glucose into usable energy. Cellular respiration starts with glycolysis, the breaking of sugar taking place in the cytoplasm. The first half of glycolysis is known as the energy investment phase, where sugar breaks and produces 2G3P. The second half of glycolysis is the energy payoff phase, where the G3P is transformed into pyruvate. These reactions are powered by two inorganic phosphate atoms leaving the G3P during glycolysis and binding to 2-ADP, effectively creating 2-ATP. This reaction makes two other ATP, meaning that the whole reaction of glycolysis results in 4-ATP. The electron carrier NAD+ then takes two electrons, reducing it to NADH, that come from the release of phosphate during the reaction of G3P into pyruvate and transports them to the mitochondrial matrix. This is the beginning of the link reaction, also known as pyruvate oxidation. Pyruvate oxidation begins with coenzyme A bonding to pyruvate, which then forms acetyl-CoA. This process produces an NADH. Next is the Krebs cycle. The Krebs cycle takes the acetyl-CoA made during the link reaction and binds to oxaloacetate, a 4-carbon molecule. CO2 is released during this reaction, along with 3 NADH and 1 FADH2 being produced. Then 1 ATP is produced for every half molecule of glucose, or 2 for every complete glucose molecule. The ATP is formed due to the release of energy from the two carbons separately breaking off from the citrate, which then causes an inorganic phosphate to be attracted to N2 and bind to ADP. Both the link reaction and Krebs cycle happen twice for every molecule of glucose. The next stage of cellular respiration is oxidative phosphorylation. Oxidative phosphorylation begins with the electron carriers and NADH and FADH2 that were produced during the oxidation of glucose. These carriers drop electrons and hydrogen atoms off at the inner mitochondrial membrane. The hydrogen atoms head to the intermembrane space while the electrons begin heading through the electron transport train, a series of membrane proteins. As the electrons travel through the ETC, they make their way to the final electron acceptor, oxygen. These electrons attract hydrogen atoms from the cytosol that then form water when bonded to the oxygen. The hydrogen atoms that were dropped off by the electron carriers begin to form a proton gradient, which is what powers ATP synthase during chemiosmosis. The hydrogen atoms from the intermembrane space cause ATP synthase to spin, and as it spins, ADP and inorganic phosphate come together to form ATP. This process is where most of the ATP produced come from. If working at full capacity, which is very rare, oxidative phosphorylation produces up to 28 ATP molecules, meaning that roughly 32 to 34 net ATP are produced during both glycolysis and oxidative phosphorylation. As previously stated, most organisms perform cellular respiration, but there are some that do fermentation instead. Fermentation is the same as cellular respiration, but it does not continue after glycolysis. The purpose of fermentation is to oxidize NADH into NAD+ so that glycolysis can continue. The NADH that picks up electrons after glycolysis bind to other molecules, forming either alcohol or lactic acid. The difference between alcoholic and lactic fermentation is the production of CO2, which is not produced during lactic acid fermentation. Now we're moving on to photosynthesis, a similar reaction to cellular respiration, but one that happens in plants. There are two parts to photosynthesis, light-dependent and light-independent reactions. The light-dependent reaction takes place in the thylakoid membrane and the light-independent in the stroma. The electrons are able to move due to the light, which then has an effect on how animals see color. What colors animals see depends on what pigments are in the organism that we are looking at and the wavelength of the light. The pigment in plants that allow us to visualize plants as green is called chlorophyll. This means that all light energy will be absorbed except for what we see as green. That light is reflected, allowing us to see what we do. In LDR, sunlight goes into photosystem 2, an enzyme, which then catalyzes the breaking down of water into oxygen and hydrogen. This happens many times. The oxygens then bind together to make O2, which is released into the atmosphere. The electrons released from the breakdown of water travel through an electron transport chain to photosystem 1. When in photosystem 1, light is reabsorbed by the electrons so that they are able to continue moving through the membrane until they reach NADP+ reductase. NADP+ reductase catalyzes the reduction of NADP+ into NADPH with the addition of electrons and hydrogen from the previous reactions. At the same time, the breakdown of water and the movement of electrons leads to the creation of a proton gradient, which ATP synthase uses to catalyze the production of ATP. The NADPH and ATP produced by the light-dependent reactions are involved in the other portion of photosynthesis, light-independent reactions, aka the Calvin cycle. The Calvin cycle starts with a 5-carbon sugar ribulose biphosphate, RuBP. Rubisco, an enzyme, and CO2 come together with RuBP, a process known as carbon fixation. This leads to the formation of a 6-carbon sugar that rapidly degrades into a pair of 3-carbon sugars, known as 3PG. The NADPH and ATP from the light-dependent reactions are used to reduce 3PG into G3P. For every 6 times this happens, 5 pairs of G3P are regenerated into RuBP, a phosphorylated form of ribulose, which then fixates with CO2. The other pair of G3P is used to create a glucose molecule, which is then used for glycolysis. The 3 RuBP that fixate with CO2 complete the cyclic nature of the Calvin cycle.